Hello girls, I hope you're staying safe at home. To continue with respiration in plants, today we will do certain experiments based on respiration in plants. So the first experiment is to prove that oxygen is used up in respiration. So for that we will set up an apparatus in this manner. There are two flasks, A and B. Okay, it is placed on a stand and raised high up. Two stands are there. Okay, and in flask A, if you see, there is a test tube hanging there with soda lime. Okay, and germinating seeds on wet cotton wool. It is placed on wet cotton wool. And in flask B, we have soda lime again, and dead bean seeds are placed here. Okay, now what does the soda lime do? It absorbs carbon dioxide which is given out by the seeds. Now in flask B, since we have placed dead beans, what will happen? There'll be bacterial action on the dead beans. So we have put wet cotton wool together with some antiseptic so that bacteria will not grow here. And because bacteria is a living organism, it will respire and it will use up oxygen. So we will not get the results. Okay, so that is why we use antiseptic. Please keep in mind. Now, what have we dipped this two delivery tubes into? We have dipped it in water. So here there is a beaker with water in it. Okay, so in the beginning, what will happen? Inside this flask, there is some carbon dioxide. Here also, there is some carbon dioxide. So as soon as soda lime is put in here, that carbon dioxide is absorbed by the soda lime placed in both the flasks. So in the beginning, you will see here the level of water has risen because some vacuum was created here. How was the vacuum created? Because carbon dioxide was absorbed. So water will be sucked up. So in the beginning, in both the cases, the water will be sucked up to this level. But after that, you will see that the water level in flask A will continue rising up, but it will not rise up in flask B. Why is this happening? Because the germinating seeds are using oxygen, and whatever carbon dioxide they are releasing is being absorbed by soda lime. So oxygen level is also falling down here. So the vacuum is being created which pulls up the level of water of the delivery tube connected to flask A. Okay, and one question if I ask you, you will be able to answer. Why is there a slight increase in the level of water in the delivery tube flask B? Because I already told you. Okay, what will happen? The carbon dioxide which is present inside the conical flask is absorbed by the soda lime in the beginning. So the level of water rises up to a certain level. And after that in flask B it will stop. But the delivery tube leading to flask A will continue rising because oxygen is being continuously used up. But in case of flask B, why is there no increase? Because the seeds that we place there are dead. So the oxygen will remain intact inside conical flask B. So in this case, out of these two, in biology, in any experiments, there is always a control. Okay, remember in the seed chapter also we did talk about the control. So in this case flask B is the control because here we have placed dead beans, showing that dead beans do not use up oxygen when they since they are dead, they will not respire. So the next experiment is to prove carbon dioxide is produced during respiration in germinating seeds. Okay, so this is a very simple experiment. We take two round bottom flasks. One is always the control. Okay, A and B, and in one we put wet cotton wool with soaked beans, and in the other boiled beans on wet cotton wool, but remember it must always contain an antiseptic. Okay, why are we using antiseptic? So that there is no bacterial growth. Okay, now which antiseptic do we usually use? I forgot to mention in the previous experiment also, we use carbolic acid, please remember. Okay, so that bacterial growth does not take place. Otherwise what will happen? Bacteria, they are living organisms and they respire and they release carbon dioxide. So then we secure the mouth of the round bottom flask with a cork. Okay, then after a few days we will see that the seeds in flask A will germinate and flask B will not germinate because they are dead. Okay, and after that the gases in each flask are tested by removing the cork and tilting it over lime water. So I think you can tell me the result. What result will you see? When we tilt flask A, the lime water will turn milky, but when we tilt flask B, what will happen? Lime water will not turn milky because there is no carbon dioxide inside this flask. Okay, now carbon dioxide, remember children, is a heavy gas and it will easily drop down. Next, moving on to a similar experiment to show that carbon dioxide is produced during respiration of germinating seeds. So this is what you have to do. Okay, there are 4 conical flasks, A, B, C and D. Okay, and see the arrangement. From outside, the air is coming through this delivery tube and it is dipping inside potassium hydroxide solution, soda lime. What does it do? It absorbs carbon dioxide. And after that, it is passed through lime water. This lime water will not turn milky. Why? Because the air which came in went through the potassium hydroxide solution here and all the carbon dioxide was absorbed here. So the air which comes out from here and goes into flask B does not contain any carbon dioxide, so the lime water will not turn milky. Now the air will continue going and it goes into the conical flask C, which has germinating seeds, and germinating seeds, you know, respire, so there is lots of carbon dioxide given out by the germinating seeds in this flask. So the air which goes out from here goes into the last conical flask D, which has lime water, and this lime water turns milky. So from where did the carbon dioxide come? Not from outside, not from air, because whatever came out from the air got dissolved in this potassium hydroxide solution. This lime water did not turn milky, so the carbon dioxide came from respiration by germinating seeds. So that is why the lime water turned milkier. And finally, to suction air pump means from here it is being pulled, air is being sucked out by means of a suction pump. So that is why air is coming from outside and then it is going in through each and every conical flask. I hope it is. So till now we had discussed experiments where germinating seeds were taken. Now this experiment is to prove that green plants give out carbon dioxide during respiration. Okay, so this is how we set up the experimental setup. There is a potted green plant here at the middle, and there is a bell jar. This is a bell jar which is inverted over it. Okay, the bell jar has a cork in it through which 2 delivery tubes are coming in. Okay, here and here. Now one delivery tube is coming from conical flask A, and into conical flask A, air is coming from outside. Okay, it is coming from outside and it is passing through soda lime. By now you know, children, what does soda lime do? It absorbs the carbon dioxide. So the air which passes through soda lime, all the carbon dioxide is absorbed and it passes through lime water. The lime water does not turn milky. Why? Because the air does not contain any carbon dioxide because the carbon dioxide was absorbed by soda lime, which was placed here. So the same air goes into carbon dioxide free air, goes into the bell jar where the green plant is placed. Now the green plant respires and gives out carbon dioxide. It takes in oxygen. So the carbon dioxide containing air will pass through this delivery tube. It goes into conical flask B, which has lime water again, which turns milky. And air suction pump, I already told you, it is air from outside, right from here. Air which is coming from this first delivery tube is pulled in through the suction pump, which is attached to the delivery tube, which is going right out. So this clearly proves that the carbon dioxide which came from A did not contain any carbon dioxide because the lime water in flask A did not turn milky, but the lime water in flask B turned milky. Why? Because the air which came out from the bell jar having a plant in it, the plant respired and gave out carbon dioxide, which turned the lime water in flask B milky. Okay, now next experiment is to show that heat is given out during respiration. This is also a very simple experiment. So this is how you set up the experiment. Okay, we take a thermos flask. You know what a flask, where we keep hot water. It keeps hot water hot and cold water cold. We take two thermos flasks and we fill the thermos flasks A with germinating seeds, cover it with wet cotton wool so that the germinating seeds keep germinating, and we insert a thermometer for the reading. You know, the thermometer gives the temperature. Similarly, we take another thermos flask B where we put dead seeds, boiled seeds, and of course with antiseptic. By now you know why we are using antiseptic, to not to allow any bacterial growth on the dead seeds, because again, if bacteria is there, bacteria will respire and again heat will be produced. So similarly, we put in another thermometer here. So after a few hours, you will see the thermometer in flask A will show a higher reading than the thermometer in flask B. It will not show any rise in temperature. Why? Because the germinating seeds in thermos flask A were respiring and they gave out heat during respiration. So before I move on to an experiment to demonstrate anaerobic respiration, children, the assignment for this week is do two experiments. First is experiment to prove that oxygen is used up in respiration, and the other one is experiment to show that carbon dioxide is given out by green plants. Okay, that is experiment 1 and experiment 3. You explain the experiment in your own words. Please do not copy from the text and draw the diagram as well. So moving on to an experiment to demonstrate anaerobic respiration. Anaerobic respiration, you know, children, respiration in the absence of air or oxygen. So we take a beaker, okay, and fill it with mercury. Mercury is a liquid metal, so there will be no air spaces in between. Then we take a test tube which is also full of mercury, so there is no air inside the test tube, and then we invert it over the beaker containing mercury. Then what do we do is we take some seeds, which is peeled pea seeds. Okay, about 8 to 10 you take, you peel it. Peeling it will make it more respiration will take place. So we peel it and then we slowly tilt this test tube and put in the peeled seeds through here. The peeled seeds, since it is light, it will go and rise up to the top of the test tube. Okay, and after a few days, about 2 to 3 days, you will see the level of mercury come down in the test tube, and there is empty space here. What is this empty space? It is carbon dioxide. From where did carbon dioxide come? From the germinating seeds in the absence of air. So what took place? Anaerobic respiration. Okay, now to test whether carbon dioxide is the gas present in this empty space, what do we do is we will take few sticks of potassium hydroxide. Remember, potassium hydroxide absorbs carbon dioxide. So we tilt it here from here. We put a few pellets of potassium hydroxide, which is light. It will rise up here. As soon as it reaches the top, what does the potassium hydroxide do? It will absorb all the carbon dioxide, and zip, immediately it will turn like this again. Because carbon dioxide which was formed here in this empty space will be absorbed by potassium hydroxide, proving that the gas produced in this empty space was carbon dioxide. And from where did it come? It came due to the anaerobic respiration by the germinating seeds. Now, if you need a control experiment, we can set it up in this way also. We can take some dead bean seeds by boiling them. Okay, and then we have to sterilize them with some antiseptic, and we put a similar setup, and what will we see? We will see that no gas will be liberated. So that is your control. Okay, now children, respiration and photosynthesis, you know that when we look at respiration and photosynthesis, they are more or less opposite to each other. Okay, so the requirements of one are the products of the other. Now for photosynthesis, what is required? Carbon dioxide. And what does it give out? Oxygen. But for respiration, what is required? Oxygen. And what is given out? Carbon dioxide. Okay, so this table shows you the difference between photosynthesis and respiration. So photosynthesis requires chlorophyll. Respiration occurs in all living cells, and photosynthesis requires light. Respiration does not require light. Photosynthesis uses carbon dioxide and water, and respiration uses oxygen and glucose. And oxygen is released in photosynthesis, whereas in respiration carbon dioxide is released. And in photosynthesis, light energy is turned into chemical energy, which is stored in the body. And in case of respiration, chemical energy is converted into heat and useful energy for different activities. And photosynthesis, it results in a gain of weight because food is produced. Okay, whereas in respiration there is loss in weight. Why? Because the food is broken down. And in photosynthesis, glucose is produced, but in respiration, glucose is broken down. So photosynthesis is also known as constructive or anabolic process. It is anabolism. And respiration is destructive or catabolic process, also known as catabolism. Now, when we compare respiration in plants and animals, okay, it is more or less the same because both are living organisms, both are living things. So there are some slight difference between plants and animals. For example, in plants there is no gaseous transport. That is, the respiratory gases just go in and out through the stomata and then from one cell to another cell. But in animals, remember, the blood transports oxygen and carbon dioxide, and one of the end products of anaerobic respiration in plants is ethanol or ethyl alcohol, but in animals it is lactic acid. The end product, one of the end product of anaerobic respiration in plants and animals, you can see it is different. And in plants, the process produces very less heat when you compare it with animals. So children, with this we come to the end of the chapter respiration in plants. So you know what your assignment is. So till we meet again, children, stay safe. God bless you.